last Monday, sorry. Monday, we finished conversation with the discussion, quick discussion of mechanical digestion in the stomach. And actually, what we approach now is the regulation of the stomach activity. I told you before that the main regulatory stimulus for the digestive system is presence or absence of food in various instances. So the activity of the stomach, the regulation of the digestive activities in the stomach, is divided broadly into three phases: the cephalic phase, the gastric phase, and the intestinal phase. What does that mean? Cephalic phase refers to the regulatory responses that stem from brain, somehow connected to the brain, CNS, let's put it this way, CNS. Gastric phase refers to the stimuli and responses that arise in the stomach, so how stomach regulates itself. Does that make sense? And intestinal phase refers to intestinal responses that regulate stomach activity. Is that clear? That separation of three phases. Okay. Now, stimuli, the events, can either increase the activity of the stomach or decrease the activity of the stomach. So they can be stimulatory or inhibitory. Does that make sense? Are we clear? Good. As I mentioned before, we always are talking about food or the process of food consumption. So if we start with the cephalic phase, what would be stimulus that would increase the activity of the stomach? Sensations of food, taste, smell, even thought. Now, taste, smell, thought of food. Are you conscious about it? Do you know that you smell food and not something else? Yeah, so that will involve cerebral cortex. Does that make sense? And in parallel, it will involve structures of diencephalon and brainstem, hypothalamus and medulla oblongata, either directly or via cortex. But the important thing is, we're talking about CNS stimulation here. Does that make sense? So you have external stimuli, sight, thought, smell, taste of food, which you consciously perceive, and they are mediated, they go through hypothalamus and medulla, which then, via vagus nerve, stimulate the secretory activity of the stomach. When we say secretory activity of the stomach, what do we refer to? When we say stomach secretion, what are the main components? Which contains of acid and pepsin and gastric lipase. But pepsin is the big time thing, okay? So, enzymes and hydrochloric acid. Does that make sense? So, when you see, smell, taste food, that immediately sends the signal to the stomach via vagus nerve to increase activity in the stomach, right? Does that make sense? Is that understood? It prepares the stomach for digestion. Food isn't there yet. It's about to. What would be the opposite? No appetite. There's no appetite. Not necessarily the absence. Well, essentially in the absence of food, there is just no stimulus. But when you have no appetite, you definitely are not going to consume any food. Then the digestive activities of the stomach, secretory activities of the stomach, are going to be inhibited. Does that make sense? So you understand the difference between stimulatory and inhibitory events in the cephalic phase. You see, the cephalic phase largely works through the CNS, through the brain or through the brainstem and diencephalon. Okay? Now, in the stomach, when food is in the stomach, obviously, presence of food in the stomach, how will it affect secretory activities? In general, without even looking there. It will increase them. We agree on that? Now, how? What's the mechanism? Distension. Distension of the stomach stimulates mechanoreceptors. Okay? Stretch receptors. And it happens, there are two reflexes. Local reflexes. What particular structure will they involve? Gut-brain, yeah. Remember gut-brain. Gut-brain essentially is the set of plexuses, the neurons, that can be found in the walls of the stomach. Stomach, intestine, in this case we're talking about stomach. We're good? Second, so-called vagovagal reflex. Why we call it vagovagal reflex? Any idea? Exactly. So, if we have stomach, okay, and we have sensory input, distension, okay, then via vagus nerve, it will reach medulla oblongata. Medulla will produce motor response via vagus nerve. That makes sense? Which will lead to increased stomach secretion. Does that make sense? Sorry for the quality of drawing. But you get the idea. So you see, again, you see a typical reflex. You get sensory input, transmission, integrative step, output, motor response. Does that make sense? Now, what would be the event that inhibits the secretion in the stomach? Think about it. Not just absence. What is the result? When stomach produces pepsin and hydrochloric acid, which chemical parameter changes in the stomach? How? It becomes more acidic, becomes lower. So when pH in the stomach becomes really low, like lower than 2, low pH is sensed by the chemoreceptors. G cells are inhibited. This is inhibition. G cells are inhibited. Lower gastrin production, low histamine levels, low hydrochloric acid production. So pH bounces back. Does that make sense? Do I understand that inhibitory mechanism here? Got it? Now, if really low pH will inhibit gastric secretions, what would increase them? High pH. I mean, I'm not saying you're going to drink bicarbonates to increase, secretions, but the idea here, when your food enters the stomach, what happens to the concentration of hydrochloric acid in the stomach? At the moment, so imagine you have a stomach with a certain amount of hydrochloric acid there. You eat something, and that something, say bread, gets in the stomach. Right at this moment, what happens to the hydrochloric acid concentration? Concentration goes down because you dilute the acid with food. Does that make sense? pH goes up. G cells sense the elevated pH and start producing gastrin. Gastrin, as we discussed before, stimulates histamine. Histamine stimulates parietal cells. Parietal cells produce hydrochloric acid. Does that make sense? So you have to understand that idea about lower acidity, if acidity decreases, if it's not as acidic, then production of hydrochloric acid will go up. It is perceived as, oh, we've got food. If acidity goes up, if there's too much acid, then secretory cells will not produce, parietal cells will not produce hydrochloric acid, and this whole process ceases. That makes sense? We're good? Now, what else will inhibit stomach, sort of directly? Sympathetic system. You can remember all the time, any sympathetic stimulus will inhibit digestive activities. We have discussed it in excruciating amount of details in AP1. I believe all of you have discussed it before in AP1. Pretty much when you are stressed or when you are exercising, that leads to inhibition of digestive activities. This is one of the reasons why it is not recommended to exercise shortly after the meal, because digestive activities in the stomach will be inhibited and you will suffer from constipation or indigestion or heartburn. Does that make sense? Questions about the gastric phase? Okay, intestinal phase. Intestinal phase is somewhat tricky. I'm going to start with actual inhibitory response, if you don't mind, because it's sort of more straightforward. You have I'm going to use this slide again. So you have a stomach, right? Very, very basic picture of the stomach, to say the least. You have pyloric sphincter, and you have duodenum, okay? So far so good? Awesome. So, a little bit of Kim, that digested food, enters duodenum. Now, for a second, let's make this whole thing anthropomorphic and think, what would you do if you would be duodenum? Would you increase gastric emptying, or would tell stomach, wait, slow down? Let's say slow down, like in the long-term perspective, slow down. Because whatever is in here, that, say, bolus, okay, this bolus needs to be completely digested and absorbed before other chyme from the stomach enters duodenum. So, chyme from the stomach, when it enters duodenum, which effect it will have on the stomach, inhibitory or stimulatory? Inhibitory. Does that make sense? In the I want to highlight, in the long-term perspective. What do I mean by that? So there's a reflex called enterogastric reflex that works through the medulla and through the gut-brain as well. Duodenum distends in response to the chyme entering it. This distension sends the signal to the stomach to be inhibited. Also, food that used to be in the stomach is partially digested. Am I clear? This partial digestion, what is it? The food is broken down to amino acids and lipids and fatty acids and so on and so forth. Does that make sense? those chemicals, mainly fatty acids and amino acids, will be sensed by chemoreceptors and will inhibit too. Finally, there is a chemical, very simple chemical, that is carried over from the stomach to the duodenum. It's very common in the stomach, not present in the duodenum. Very simple chemical. Staples of the stomach digestion. Hydrochloric acid. Stomach is supposed to be slightly, intestine is supposed to be slightly basic. When acid enters into the intestine, and it will because it's a component of a chem, does that make sense? Then acid will signal through the chemoreceptors to inhibit stomach activities. Does that make sense? So you have three components, sort of trifecta. Mechanical distension, food chemicals, like amino acids and fatty acids, and hydrochloric acid from the stomach. All these three signals, all these three stimuli in the intestine will initiate the inhibitory reflex that will slow down stomach secretory activity. That makes sense. Another mechanism for practically the same stimuli, acid, amino acids and fatty acids, and hydrochloric acid, amino acids and fatty acids, and distension, will lead to the release of hormones, so-called enterogastrones. And of those enterogastrones, two most important, I personally would say. Secretin and cholecystokinin. We're going to talk about those in details. CCK and secretin. These hormones inhibit stomach digestive activities. So to kind of summarize the intestinal phase of the gastric regulation, chem that contains acid, hydrochloric acid, nutrients such as amino acids and free fatty acids. And this chem also distends intestine. Those three stimuli, two chemical stimuli and one mechanical, they will inhibit gastric digestion, either through the reflexes, through the nervous system, either local reflexes or reflexes that involve medulla oblongata, or, not or, but and, those three stimuli will lead to the release of hormones. Secretin, cholecystokinin, vasoactive intestinal peptide, VIP. Those three hormones will inhibit stomach digestion as well. Does that make sense? As I mentioned, what we just talked about, that inhibition is a long-term effect. In a very, very, very short term, all the same stimuli, when they just start getting into the intestine, hydrochloric acid, nutrients, and mechanical stimuli, when they just start to develop, they initially release gastrin in the blood. Does that make sense? So essentially, it's something like intestine almost does this. It says in the very beginning, oh, give me more, give me more. But when the amount of chem becomes, significant, effects of the reflexes and enterogastrones overcome the effects of gastrin. And at a certain point, they shut down the gastric emptying and gastric secretory activities. Close pyloric sphincter, and, food remains in the stomach. Does that make sense? Go ahead. That's a great question. So there are two components to it. One is mechanical, feeling of satiety. That is linked to the pretty much size of the stomach. This is why people who constantly overeat, significantly overeat, volume-wise, they will have distended stomach, and they can consume more food without feeling full. Part of the reason, I forgot the name of the surgery. Bariatric surgery, when the size of the stomach is surgically decreased. So they feel satiety earlier. Does that make sense? Second mechanism is hormonal. As far as I remember, when triglycerides hit adipose tissue after you ate, first, kind of triglycerides, say the triglycerides or insulin, I'm not really sure, but the point is, after the meal, adipose tissue starts to produce leptin. Leptin acts specifically on hypothalamus and gives you conscious feeling of satiety. For instance, if you would eat protein produces leptin much faster. So we'll have feeling of satiety from the smaller amount of food. As far as I remember, carbohydrates don't do that as fast. So you need to eat a lot of sweets to actually feel full. And as I mentioned before, it's the only hormone that we can consciously overcome. Okay? But that's a good question. We have two mechanisms. So regulation of satiety comes from the stomach, mechanically, and from the adipose tissue. Going back to the intestinal stage. Are we good about, that? Oh, God, what's the word? Yeah, the long and short-term effect. So if I would ask you, presence of Kim in the intestine in the long-term perspective, how would it affect the secretory activity of the stomach? It will slow it down, inhibit it. Does that make sense? Now, this is presented here, the same idea presented on the right panel. Is the flow chart, the response. So you have Kim in the duodenum. It's fatty, it's acidic, it's hypertonic, so there's a lot of salt. Okay? So you have stretch receptors and chemoreceptors that induce reflexes. Okay? Those reflexes will decrease contraction of the stomach. It will decrease gastric emptying. And the same effect will be observed when enteroendocrine cells in the duodenum produce secretin and cholecystokinin. Gastric emptying and gastric secretory activity will slow down. I use the phrase gastric emptying often. Do you understand what I mean by that? Okay, gastric emptying. Food goes from the stomach to the duodenum. That's it. That's gastric emptying. Stomach tries to empty itself. Duodenum tries to digest whatever gets in there. It doesn't let stomach to dump everything in the duodenum all at one time. Good? The fun stuff. Exotic reasons for gastric ulcer, which you can see here. The lesion. The exotic, it's pretty much everything is gastric secretions. Too much acid. There's a tumor that overproduces gastrin. Too much gastrin, too much histamine, too much hydrochloric acid. Hydrochloric acid kills epithelial cells faster than they can recuperate. Tumor. Ulcer, sorry. Does that make sense? The most common cause of gastric ulcer is bacterium called Helicobacter pylori. I say here associated, not really associated. It causes it. So these two fellas, Marshall and Warren, I never remember the names. Those of you who are or were in my microbiology class know that I am absolutely terrible with names. So I don't remember. One of them is Robert. That's all I know. And I don't even remember who is the physician and who is the scientist. They work together. They are from Australia. They noticed that patients with gastric ulcer had biopsies positive for that enigmatic microorganism Helicobacter pylori. For several years, they tried to grow microorganism in the culture. They couldn't, until they figured out that it's not a complete anaerobe and not a complete aerobe. It's in the middle. So you have to provide just right amount of oxygen. They figured what amount of oxygen and they accumulated. And it grows pretty slow. They finally got several bottles of bacterial culture, but they still had no idea what to do with it, because mice apparently didn't develop gastric ulcer in response to Helicobacter pylori. And then one of them, I don't remember. Oh, Marshall. Okay, Marshall. You see, you can trust it. I took it from the book. So Marshall drank a beaker of bacterial culture, which by itself is a daring exercise because it stings. So a few days later, he developed classical symptoms of gastric ulcer. His colleagues biopsied his stomach. Biopsy was just textbook example of gastric ulcer. Of course, it was N of 1, but they confirmed causative relationship. So basically, what it means. Does it mean that if you don't have Helicobacter pylori, you're not going to have a gastric ulcer? No. You still may, due to different reasons, but chances are you probably won't develop it. If you have H. pylori, you probably will. Depends on, again, how many cells, microbial cells you have in the stomach. But to a large extent, like if you have it, it's practically guaranteed. And the best proof that it causes gastric ulcer, previous treatments of gastric ulcer involved mostly the acid adjustment, like proton pump inhibitors, even things to just neutralize acid, like magnesium or whatever, magnesium sulfate or magnesium hydroxide. It did not work. Only when those proton pump inhibitors like omeprazole were combined with, say, tetracycline, then it started to work, because you treated the symptom, ulcer, and or high acidity, and you treated the bug. Does that make sense? So it's absolutely treatable. It's curable, more than that. It can be life-threatening if the ulcer is penetrating. There are situations when ulcer penetrates the wall of the stomach. My high school classmate, he died of the penetrated ulcer. Took him 24 hours. He was hospitalized. The next day he was dead. It's life-threatening condition, and it's really hard to treat because all the content of the stomach essentially spills into the peritoneal cavity. Other than that, another funny thing about gastric ulcer, it's the risk factor for gastric cancer. Very well documented. People who have active, like sick, have gastric ulcer now, they more likely to develop gastric cancer later in life. If it's treated, then of course, probability decreases. What else? Helicobacter pylori is negatively associated with asthma, which means kids who have H. pylori are less likely to develop asthma. So kind of, it's your choice, either asthma or gastric ulcer, whatever you prefer. Another interesting thing about the stomach secretions is non-steroidal anti-inflammatory drugs. Aspirin, ibuprofen, acetaminophen. They inhibit basically pathways that involve prostacyclins, prostaglandins, chemicals of that certain group. We mentioned them en passant when we talked about hormones. Remember in the beginning I said that there's a group of hormones, fat-soluble hormones called eicosanoids. We never really touched on them. So those eicosanoids are important regulators of inflammation, and they regulate histamine release. Okay? So another, what they regulate bicarbonate and production of mucus. When you take inhibitors of prostacyclins and prostaglandins, production of mucus decreases, epithelial cells of the stomach become less protected, and you experience the symptoms of gastritis. That is why, when there is an aggressive treatment for pain, like pain medicine, usually things like diclofenac or ibuprofen, they are not prescribed for longer than 5-7 days. And even in this case, doctors will tell you, please monitor your stomach closely. If you experience symptoms of gastritis, go to the hospital right away. Okay? Does that make sense? With that said, we move to duodenum. It is a very short fragment of human digestive system. Duodenum comes from the Greek expression saying 12 fingers. So it's basically like this is 10, add a couple more, that's how it's supposed to, how long it's supposed to be. 12 fingers. Okay? Not this. Not this 12. It's going to be too long. This is the most important absorptive part of your alimentary canal. So you can live without large colon. You can live without huge chunks of the small intestine, huge chunks of jejunum or ileum. Without duodenum, that's going to be a crappy life. Okay? Three very important organs are connected to it. It's liver, gallbladder, and pancreas. We will talk about ducts and duct system, but if you would look at the image here at the bottom, you will see that there's a duct coming from the liver and duct coming from gallbladder. They merge together and form a bile duct. Bile duct fuses with the main pancreatic duct. And the structure here that is called hepatopancreatic ampulla. Or, again, names. I don't know. Ampulla of... There's dude in which honor this ampulla was named. Ampulla of Lodi or something. I don't remember. Anyway. Hepatopancreatic ampulla. And it makes perfect sense. Hepatopancreatic. Does that make sense? So bile and pancreatic secretions, they actually mix before getting into the duodenum. Under parasympathetic control, vagus nerve. Look, with parasympathetic, it's really easy, Unless you're talking about urinary bladder and reproductive organs or rectum, like really, really low, really in the pelvic cavity, safely assume everything else is under vagus. Seriously. Heart, all of it is vagus. Which kind of sucks because if somebody experiences vagectomy, the consequences are pretty dire, Splanchnic nerves from sympathetic, so sympathetic system inhibits, parasympathetic stimulates. Got it? Awesome. Superior mesenteric artery carries the blood to the walls of duodenum. We'll talk about, veins that take the blood away. So you're going to find a lot of folds. Why do we need folds in the duodenum? Increased surface area, increased absorption. Very approximate comparison. But imagine you have a 10-inch hose that is about 1.5 inches in diameter. Okay? So you cut this hose along and lay it flat. It's going to be size of a fairly large handkerchief. Not even a bandana. Pretty large handkerchief. And I think, okay, the volume of Kim that enters that hose, which is duodenum, is about 3 milliliters. If I spread these 3 milliliters over a large handkerchief, the layer is going to be pretty thin, right? Yes, but it's not thin enough. Duodenum has 3 major structures that increase its surface area. 2 of them are noticeable, macroscopic. Circular folds are number 1. Okay? Can you see circular folds? They circular. Okay? They have 2 functions. First, increase surface area, and 2, they direct Kim in that spiral fashion. Like in the water park, some of those water slides, they direct you in a spiral fashion. Like a toilet bowl. Okay? Does that make sense? Why spiral fashion? Why just not let Kim pass through directly? When it goes like this, it takes longer. Very good. More time for absorption. Very good. Does that make sense? Another structure that pretty much can be noticed with the naked eye, villi. Just villi. Each villus is about maybe 1 millimeter long. If you would take duodenum from human and open it up, you will see that the surface is velvety because of the villi. Does that make sense? In many intestinal diseases, and villi can be found throughout the small intestine. We actually, when we're going to be talking about small intestine, we will be focusing entirely on the duodenum. Jejunum and ileum are important, and anatomically, they're not very much different. The reason why we're focusing on duodenum is because that's the place where Kim is mixed with pancreatic juice and bile. And that's the place where most digestive and absorptive activities happen. Does that make sense? So, villi give duodenum velvety appearance. In diseases like colitis, Crohn's disease, inflammatory bowel disease, villi are gone. Actually, it gives intestines, those diseases, especially Crohn's, gives intestines very characteristic appearance. Cobblestones. If you look under the microscope, it's cobblestones. Inflamed patches look like cobblestone. I have to make a disclaimer. The reason why I know that, my wife works in the lab that studies Crohn's disease, so it's our dinner talk often. But it's fascinating, and there's some things that they study mostly, they have some human samples, they study mostly in mice. I will refer to some mice studies. So villi, you can see them here. Essential in this model. That's great. So this is model, right? These pink things that look like wieners, okay? Those are villi. That's a cross-section of the villus. Does that make sense? Inside of the villi, what you're going to find? You can refer to the model and you can look there. So if you look at the villus, you can see blood vessels, venules and arterioles. Venules are shown in blue, arterioles are shown in red. See that? When we say venules and arterioles, it means small veins and small arteries. And you can also see what's shown here in green. There's green vessels. Green here. Those are called lacteal. Lacteal carries lymph. It's a lymphatic vessel. And the function of lacteal is absorption of fat. It's a main function. Good? Got it? We didn't talk about lymphatic flow, but let me tell you, lymph is generated in the tissues. And from the tissues, it enters lymphatic vessels, and lymphatic vessels will collect the lymph eventually, and they will dump. There's a thoracic duct and main lymphatic duct, as far as I remember. Anyway, they will dump the lymph into the subclavian arteries. Subclavian veins, I'm sorry, right here. So whatever fat will get into the lacteals, these lacteals will carry it with the lymph, okay, to the bigger and bigger lymphatic vessels. And eventually, that fat from the lacteals will enter the circulation, will enter blood, and will be delivered to the liver tissues wherever it needed. Does that make sense? If you ask me why not blood, I answer, I have no idea. Really. It works. I cannot think of can't think of the selective pressure. The only thing I can think of is maybe the problems with the diffusion across the cell. Lymphatic vessels have slightly different structure. They have very permeable walls. So maybe for the fat, it's easier to get into the lacteal. Okay? In the lamina propria, you're going to find, well, you can see it in the image. Not so much, no, not on the, so this white stuff here, this white stuff, that's lamina propria. In the lamina propria, there's going to be mucosa-associated lymphoid tissue, MALT. Mucosa-associated lymphoid tissue. It's one of the first barriers that protect you against pathogens entering from the intestines. Am I clear? So far we've got circular folds. We got villi. Inside of the villi, we figured there are blood vessels, lymphatic vessel, called lacteal. Lamina propria carries the lymphatic tissue, lymphoid tissue. Now each villus is covered with the layer of epithelial cells. This model, you can see the layer as the pink covering. So that pink covering represents epithelial cells. In these images, you can see this is the layer of epithelial cells. This is the layer of epithelial cells. Does it make sense? Each epithelial cell has microvilli. Microvilli, which you can see right here, microvilli or brush border, are folds of the membrane. They aren't protein projections. They aren't, carbohydrate projections. They folds of a membrane. Imagine that you take a surface of the cell membrane, and fold it. Okay? The only downside of me showing you this is that it's not folded like this. Those are really spiky finger-like projections of the membrane. Does that make sense? That's why they're called microvilli. Okay, so they create this surface like a forest, like toothbrush. Does that make sense? So there are three structures, turns out to be, in the intestines that are responsible for the increased surface area: circular folds, villi, and microvilli. Go back to our talk about size. From the large handkerchief, we now go to the real surface area of the duodenum, which is about 15 square yards. Now imagine you spread 3 milliliters of Kim over 15 square yards. That's going to be a very thin layer. That's going to be a layer on the molecular level. Does that make sense? That increases largely, enormously facilitates the absorption. We good? That makes sense? Another, another important feature of this brush border, that's the place where a lot of digestive enzymes sit. And that kind of makes sense. Think about this. Nature's kind of stupid sometimes and inefficient. But in this case, what happens? If you throw digestive enzymes in the intestinal lumen, you're going to lose them. They're going to do some function, right? But they're probably going to be destroyed by other digestive enzymes. Do you understand what I try to say? So instead of putting them in the lumen, these enzymes are sitting in the brush border, and when semi-digested nutrients enter the brush border, and they will, those enzymes digest them, completely degrade them. Does that make sense? The crevices on the surface of the intestinal mucosa, called intestinal glands or intestinal crypts, and they produce secretions involved mostly, mucus, alkaline mucus. That makes sense why it's alkaline? Because you have just acid coming from the stomach just now. There are absorptive cells. This is a type of the cell which I will not ask you about. You know what I'm saying? I will not ask you a question, what is the function of absorptive cell? Enteroendocrine cells. This produce hormones that we talked about already, cholecystokinin and secretin, and Paneth cells. Paneth cells are sort of resident macrophages, They can phagocytose pathogens and destroy them. Before we move on, any questions? Know the functions of the cells, please. So I want you to know the surface, the structures that increase surface area. Understand the idea of brush border and enzymes sort of sitting in there. And those cells. It can be found in the epithelium. Yes? No. Let me try to clarify it for you. Sometimes, dynamic drawing works better. This is villus. So it's macroscopic structure. On the surface of it's not to scale, okay? On the surface of the villus, there are cells. They're not complete yet. The picture is not complete. Columnar cells. on the surface of the columnar cells are microvilli. So far, do you follow me? Now, blue dots are going to be digestive enzymes. Digestive enzymes are in the membrane of microvilli. So far, do you follow me? Kim, food is here. Does that make sense? Blood is here. Does that make sense? Did I answer? So those digestive enzymes are sitting in the membrane of the microvilli, like in, microvilli is the membrane. So they're sitting in that membrane. They're not going to the lumen. They're not going to the blood. They're sitting there waiting for those food particles. It's not food, like molecules to come to them to be destroyed. Did I answer? There's a gland, so-called Brunner's gland, that's located right at the junction between the stomach and duodenum. Brunner's gland produces especially thick and especially alkaline mucus. Do you see the link between the gland location and its secretion? It's at the junction with the stomach. It produces that thick alkaline mucus to neutralize the acid immediately. Does that make sense? Now, right out front, you need to remember these enzymes. I know you're going to tell me, okay, the fact that other people suffer doesn't make my life any easier. I used to have extended response questions. And one of the questions sometimes was, what are the enzymes of the brush border? So students had to write them down with a substrate and everything. What you need to know, I ask you, what is the substrate for dextrinase? And you tell me, the substrate for dextrinase are the dextrins. What are the products of aminopeptidase? You tell me, amino acids. Does that make sense? And it's going to be multiple choice. So you have to recognize, it's still memorization, but not as brutal. You won't have to list, 8, whatever. But I will try to make sense out of those. So, going back to your question. Brush border. This is electron micrograph of the brush border. So in the membrane of those microvilli that you can see, enzymes are going to be like this. That makes sense. I mean, they're too small. You can't see them really, but they're going to be there. So what they digest, those enzymes? You have 4 enzymes right here that digest sugars. Dextrinase, lactase, sucrase, and what else do I have? Maltase. Yeah, those 4. What I can ask you, the substrate. That's the simplest, really. Because the substrate for lactase is lactose, for sucrase is sucrose, for maltase is maltose, for dextrinase, dextrins. Okay? There's no sugar called dextrinose, it's just dextrin. In terms of the products, I will spare you suffering. In terms of the products, you have to say monosaccharides. All these are monosaccharides. saccharides. Does it make sense? So people that have lactose intolerance, they do not have a gene that produces lactase. Now another interesting feature, if you don't have lactose intolerance, I mean, when we're born, we eat milk. And lactose, is a milk sugar. And unless it's genetic, we produce enough enzyme, lactase, to break down milk sugar. As we grow up, often, especially like in adolescence or later, we stop drinking milk. I'm not judgmental, it's a personal choice. So people stop drinking milk, and they may still eat some of the dairy products. But the trick with dairy products is that microorganisms that ferment, they ferment lactose, so they break it down. Does that make sense? So the levels of lactose in the fermented milk products aren't as low. So even if you eat fermented products, you don't consume as much lactose as you would if you just drink milk. If you don't drink milk, if you don't consume large amounts of lactose, the levels of the enzyme called lactase go down. You still have a gene, you're fine. Your cells just do not produce it as much. And then at some point, somebody may read, multiple scientific studies about the benefits of drinking milk. There are many. Whole milk, for instance, was shown to actually prevent development of hypertension. There are large studies showing that. So they decide to pick it up. They buy a gallon of milk and they drink 2 quarts. What are they going to experience? Several fantastic hours on the toilet. The reason for that, they consume large amount of lactose. The enzymes of the brush border didn't catch up yet. If you will just start from a little bit and will build it up, if you're genetically competent, if you don't have genetic lactose intolerance, you'll be fine, eventually. It's kind of an interesting feature. If you stop drinking milk, you have problems digesting it initially. Did I make myself clear in terms of the substrate and the product? Good? Any questions about this 4? Enterokinase will be the last. Aminopeptidase and dipeptidase. You see peptidase? Peptide. So the substrate is peptide. The product is amino acids. Dipeptidase breaks down peptides that consist of 2 amino acids. Aminopeptidase can act on the longer ones, just chips them off from the end, one by one. Everybody is comfortable with the idea of substrate and product. Or crash course reminder, don't hesitate. Substrate, product, enzymatic reaction. Substrate is converted to product. Enzyme facilitates this conversion. Comfortable about that? Good. Again, for dipeptidase and aminopeptidase, the substrate is peptides, the product is amino acids. That's all you need to know. Am I clear? Nucleosidases and phosphatases. Nucleotide consists of 3 parts, which are come on. Nitrogenous base, phosphate residue, and ribose sugar. Nucleosidases and phosphatases break this whole thing in pieces. Okay? They break down nucleotides to absolutely elementary building blocks. Does that make sense? So, substrate is nucleotide, products, nitrogenous bases, pentose sugars, and phosphates. So far, does that make sense? Enterokinase. This is the only non-digestive enzyme in the brush border. Important non-digestive enzyme. So what it does, enterokinase activates another digestive enzyme that is called trypsin. Trypsin is produced by pancreas. To be exact, it's not trypsin that's produced, it's trypsinogen. When pancreas releases trypsinogen, trypsinogen has to be activated. Enterokinase does just that. Activates trypsinogen so it becomes trypsin. Did I make myself clear? So the substrate will be trypsinogen, the product will be trypsin. Good? That we're going to do it after the break. One thing that I want to briefly mention. Bacteria, well, I'm a microbiologist, so I can't just not talk about them. Are there many bacteria in the stomach? What do you think? Like variety, like millions of thousands of species? Not really. It sucks. It's acidic. It's horribly acidic. Only few can survive. Interestingly enough, in the duodenum is not sterile, but it's close to it. There are very few bacteria in the duodenum. They all hang out in the large intestine. Well, cecum and large intestine. In the small intestine, there are very few bacteria. And that is one of the reasons, if you've heard about such thing as fecal transplant, treatment of various, mostly Clostridium difficile, Crohn's disease as well, the first treatments, how they were done. Anybody knows? People drank a fecal suspension. And it worked not so well. And then scientists realized what happens. Drink a bunch of bacteria. Mouth and esophagus is fine, but then they have to cross stomach, and not everybody makes it. And then you have a whole small intestine. Not everybody makes it. And what ends up in the large intestine? God knows what's there. So what they then started to give enemas, which worked much better, but still the procedure is far from pleasant. So what they do now, you can actually donate poop if you're healthy. I think some hospitals even pay like $25. I don't know, is it by weight or... But they package it in the special, so they dry freeze it, so bacteria survive, and they package it in the special peels, which have coat. It's like time release. So it will dissolve only in the large intestine, and will release them just to the port, which is pretty cool. Okay, let's take a break.